little circle arrows. Yeah.
Alright, so let's talk about what we learned last time. Oh, first of all, profession. I am so grateful after this. I'm sorry, it did take longer. A lot of writing, more than anything, trying to give as much partial credit as I can. So I'm a big believer of partial credit. If you put an answer wrong for one part and then that answer that you put carries over to the next part, I try to give you partial credit because I don't want to keep dinging you. Like if you put one thing wrong, then I keep going, no, and I keep dinging you. So again, it just kind of takes time because of the partial credit. But I'm hoping to have them done by, what's it? I'm hoping to have them done still by Thursday. So by Thursday night. So your grades will go up. You might not get it back, but you'll at least feel. Okay, last time we were talking about metabolism. We were talking about enzyme regulation. So we said that there were two ways to regulate enzymes. What was one way that I could shut an enzyme off? Okay, so that's a way to change how the enzyme works. There were two ways I could change how the enzyme works. That was pH and temperature, right? But what about if I want to turn off the enzyme? Inhibition, and there were two different types.
Non-competitive and competitive.
Good, non-competitive and competitive. So non-competitive inhibition is when I'm binding at a different site than the substrate. What was that site called?
Allosteric site.
Allosteric site, good. And then I also can have competitive inhibition, which is I'm binding for the same site as the substrate. So those are some keys for my enzymes. Also knowing temperature and pH affect how they work, and then if I push them too far, they could be irreversibly denatured. We also talked about redox reactions. So we talked about losing electrons and we talked about gaining electrons. So if I lose an electron, what's that called?
Oxidation.
Oxidation, good. And I'm hearing more voices cheering up there. And if I gain electron, reduction, good. Yeah, that's good. I like when I hear everybody chiming in. That makes me feel really happy. So that's good. I also like that you're hearing everybody. That's good too. So don't be afraid to shut up. I like it. So again, these are some key terms. So I've had some people ask me like, how do I study? If you want to write your notes, again, you can. I would not rewrite a book. I would be careful. I would use your time valuably. Do bullet notes. Do quick notes so that when you see something like put redox and put dash, Leoger or oil rig, something that will kind of channel your head, channel your brain, so when you see that on a test, you remember. And then when you're studying, go through and talk those things out. Try to be able to explain them in more detail. Let's see, we started talking about glycolysis. So we said that glycolysis is the use of sugar. I told you that there's two different pathways that we can go down in our sugar, depending on what kind of cells we are. One of them is going to result in ATP, the other is going to result in nucleic acid production, right? So we talked about how we can make nucleotides. And we said that we also had another pathway, a third one, that is unique to certain types of bacteria. So we're going to be covering these different pathways today. The first pathway that we started with was the EMP pathway. This is the one that's the most common one that we learn in physiology. Okay, so the EMP pathway. So we said that in this pathway, we're going to break apart a glucose molecule, and we're going to end up with some ATP. What was my net total of ATP? Two. Excellent. I also ended up with some Uber drivers. What were my Uber drivers? NADH. Good. How many NADHs? Two. Good. So it's a 2-2. 2 ATP, 2 NADHs. I also end up with my glucose changing its name. It goes from a 6-carbon sugar to a 3-carbon molecule. What is that 3-carbon molecule called? Pyruvic acid. Good. Pyruvic acid or pyruvate, I don't care. They're used interchangeably. All right, so let's go through that process just to make sure that we're all on the same page. So let's make our circle. We got our glucose, right? What's the first thing we do? We open them up. What do we attach? Phosphate. What do we attach? Declan, where is your circle and where are your phosphates? Let's keep doing this. Okay, let's all go back to circle. Thank you. And we're going to open up. See, now I know your names, you're like, crap, she said pause. Okay, phosphate, phosphate, what do we do next?
Split.
We split, good. What goes here? Good, what goes here? Okay, what do we do now? We rearrange. When we rearrange, we're creating the NADHs. Okay, we're having carbons holding hands. Carbons let go of hydrogens, grab another carbon, so we get some double bonds. That hydrogen leaves, he goes and joins our NAD and becomes NADH. Okay, so we've gone through here. Now we're gonna take our phosphates off, right? So we pick 1, 2, 3, and 4, and now we end up with our 3 carbons, because we've been split. What do we call this? Pyruvic acid. Excellent. Okay, all together, one more time, let's go faster. Circle, open, split, phosphate. Oh, I'm sorry, I blew it. Let's try again. Okay, change the hands. Okay, let's go again. Okay, circle. What do we do next? Open, phosphate, phosphate, split, phosphate, phosphate, rearrange, and we're gonna do a phosphate, a phosphate, a phosphate, a phosphate, a 3 carbon, and now we end up with pyruvic acid. Everybody good with that? Okay, you got that. You just explained this. See, it's not as scary as it appears, right? That's really scary. We broke it down. Now, a couple of things. When we pick those phosphates off, remember we're generating 4 ATP, but our net total is 2. Why is my net total 2?
Because you had a sacrifice.
Because I had these 2 to get started. Yeah, I had to pay a price to play the game. Okay, so in this case, in this point in time, I've gone through glycolysis. This is what 98% of the cells go through. And the beauty of glycolysis is it's fast. It goes really fast. So we can do this very, very quickly in the cells, and we can make some simple sugar. We can make, some simple energy, 2 ATPs. We can make some Uber drivers, some NADHs. Also, as I mentioned before, no oxygen is required for this step. So that's really helpful too, because we're not dependent on making sure we have oxygen in the cells. They can do this without oxygen. The minus side, I did a lot of work. Not really getting a lot of bang for my buck. When that dance was tiring, I only got 2 ATPs. So that gets us to where I think we left off, right, which is our next 2 pathways. So our next 2 pathways are different ways that we can break our sugars apart. So I'm going to do the EMP pathway. So that's, and remember, we refer to these as glycolysis, and these are all different types of glycolysis, 3 types. And the reason that they're called glycolysis is because I'm splitting sugars. So that's why it's referred to as glycolysis. So this is the first one. This is the first pathway, which is the EMP pathway. And this is going to be eukaryotes, and I'd say probably about, I don't know, maybe 97% of our prokaryotes are going to do the EMP pathway. And again, our net total is we end up with 2 ATP, 2 NADH, and 2 pyruvic acids. Okay, so this is the one that we're going to do most often. Most of our cells are going to do this when they need energy. So I'm just going to abbreviate that as a capital E. Okay, the other pathway that we can go down, the one we're going to cover now, is the pentose phosphate pathway. We refer to this as the PPP pathway. And every cell does this. Okay, so there are no known exceptions. Every cell does this. And the reason every single cell does this is because we're going to be making nucleotides. And what are nucleotides used for? Nucleic acids, yeah. So we're going to be making nucleotides to make nucleic acids. So if I'm a cell and I want to do any kind of replication or growth or repair, I'm going to need new nucleic acids. So this is the pathway I'm going to do to get nucleic acids. I am not going to generate any ATP. This is not an energy pathway. Okay, so every cell will do this to get nucleic acids and nucleotides, but this is not an energy pathway. The other pathway that we can go down, so these are our choices, this would be 1, 2, and this will be 3, is the last one that we'll cover in here, which is the Ettner-Dwardoff pathway. And again, only about, I don't know, maybe around 3% of prokaryotes do this. No eukaryotes do this. They're smarter, at least not that we found. And then this one is going to be an energy pathway as well. We're going to find it's not that efficient for reasons that we'll talk about in a few. So when you picture glycolysis, if I say glycolysis on a test, that means that I'm we want to get specific, I will tell you the EMP pathway, the PPP pathway, or the PPP pathway. Or if I ask you which pathway and give you this information, you should be able to identify the pathway we're doing. Does that make sense? Okay. We're starting these right now, so I just want to make sure that we're all on the same page. Okay, so the pentose phosphate pathway, or the PPP, is the one that we all follow. Every cell does this, so that we can make our pentose sugars. Yes?
Under EMP, does that say that some people will be
For what's on my board? So under the EMP, it should be 97%. Yeah, about 97. And again, this is just a rough estimate. This is a Hadley estimate. I haven't seen a recent publication. I'll try to find one. But 97% of prokaryotes are going to do EMP. So most of them choose this pathway.
What does it say under the ED?
Under the ED, about 3%. Yeah, so I'm guesstimating here 97%, 3%. It might be closer to 95 and 5, but it's the idea of this is the majority. That's really what I want you to know. I'm not going to ask you percent. I just want you to know that the majority are doing this. Okay, all eukaryotes do this. Most prokaryotes do this. This is an old pathway. It's an ancient pathway. This is the flip phone pathway. This is the one that most of us have left behind. But there's those few holdouts that like their flip phones, right? Oh, we all know somebody like that. Okay, so the pentose phosphate pathway. This is where we're going to bring glucose in. So all of these pathways, again, since they're sugar splitting, they're all going to start with glucose. That's what we're starting with. I'm going to bring glucose in, but in this pathway, I'm not trying to make any ATP. Instead, what I'm going to do is I'm going to take that 6-carbon ring, I'm going to open it, and I'm going to kick 1 carbon off. And so I'm going to end up with a 5-carbon ring, okay? And that's what I need. A pentose ring is a 5-carbon, penta means 5. So a pentose ring is where I'm basically setting the basis of being able to make a nucleotide, okay? Now, when that carbon gets kicked off, it leaves us CO2. So we just kick it out of the body as a waste product. We don't need them. So we're gonna just hold on to that sugar. We're gonna do some rearrangements. And again, we're not doing this for any energy. This is just for using this as a building tool. So what's gonna happen here is we're gonna have our glucose come in. We open it and we attach a phosphate. And again, the reason we attach a phosphate is to prevent it from leaving. We put a phosphate on it to tag it. And when this happens, if you read your books or any information, it'll tell you that we created glucose 6-phosphate. What that means is that I put a phosphate on the 6th carbon of the glucose. Don't worry, I'm not going to ask you that. I'm just letting you know in case you see this reference. So when you take organic chemistry, if any of you have the pleasure of doing that, it will tell you, you'll learn that we name things based on where they're attached. So in this case, when it says glucose 6-phosphate, that means a phosphate on the 6th carbon. So we just did that, we tagged that carbon so it can't leave. And then we're gonna kick the other carbon off. Okay, and then we'll hold it back up. So what's gonna happen then is, since I am rearranging my glucose, once again my glucose is losing electrons. So if I'm losing electrons, am I getting oxidized or reduced? Oxidized, yeah, so it's gonna be getting oxidized. Now, in this pathway, we are gonna create an Uber driver, but it's not the one that we usually see. Instead of NADH that's being created, we're creating NADPH. Still an Uber driver, its job is still to carry electrons elsewhere, but it's just being used in this case to help shuttle electrons to a different place. It's not necessarily going to the electron transport chain. So if we look at this pathway, we start our dance like normal. We start with a circle, we open it up, we add our first phosphate, and then we stop. And then we take a side step, it's kind of like a cha-cha, you know, take a side step. And then you're moving over into the pentose ring. So what's gonna happen when you go over to this side is we are going to kick the carbon off the other end, and then we're gonna fold our ring back up. So what I've essentially made is I've gone from like my 6th carbon here, like this is my 6th carbon, I don't know how to do this like that, to a 5 carbon. Okay, that's really all we're doing. When we're done, the whole point of this is just to end up with that 5 carbon ring. And then and that's gonna be used for the nucleotides. No energy, we're not worrying about trying to shuttle things to electrons and proteins, it's just a simple rearrangement. Okay, that gets us to our third possible pathway, and again, this is one that we're not really going to see with eukaryotes. It is something that is a throwback, okay? Only a few bacteria do this. Like I said, it's a very small percentage. Our friend Pseudomonas, who we're gonna get to play with in lab here pretty soon, is one of the few that does it. Rhizobium and Agrobacterium, these are ones that are associated with soils. They can do it. We have one gram-positive who's known for doing it, who's Enterococcus. We'll get to play with him in lab. They're able to do this pathway, which again is really old school. Now, the idea behind the ED pathway is we start glycolysis, but our usual EMP, but they don't have enough enzymes to finish the job. So they kind of halfway do the job. So what they do is they start with the glucose ring, they open it up, they add their phosphates, things are going good, then they separate, and then they kind of just ignore this side. And they only process one side of the 3-carbon chain. So that's gonna be the side that we're gonna rearrange electrons with. That's gonna be the side that we're gonna get some Uber drivers. This other side just kind of tags along because they don't have enough enzymes to do both sides. They're basically like kind of picking and choosing. So what this means then is they get a much lower yield for ATP. Not much, but like half. So this was our process in the EMP pathway. Okay, so this is our EMP pathway that we just danced to. This is their process. So they open up, they are only going to phosphorylate one side. So they open and then they phosphorylate just one side to keep it from leaving. And then they break apart, okay? And then one side just gets ignored, and the other side, they're gonna pop one of the phosphates off, and then the second one they've added, they pop off. But what's gonna happen then is they're only going to yield a total of 1 ATP. So when they pop those phosphates off, they're only popping off on one side. That's gonna create 2 ATP, but they played one, or paid one to play. So when we walk away, we only have 1. Alright? So the idea behind this then is that when we do our dance, we process both sides. Remember how we did our fun little hands and we process both sides. So we walk away with a net total of 2 ATP. They're only processing one side. So they're only walking away with 1 ATP. Alright? They're also going to walk away with 1 NADH. So everything's like kind of halved in there. Yeah.
And it still ends up with 2 pyruvate.
It does, because of the fact that we have to open and break that chain. We're going from that 6 carbon, when we break it in half, we end up with 2 3 carbons. So they're still gonna end up with 2 pyruvic acids, but one's been processed and one not.
So that means that if it's not phosphorylated up there, it is already pyruvic acid.
Correct, correct. So it arranges right away and becomes the end product, which we call pyruvic acid. But this side gets some processing, it gets phosphates added and then pulled back off. Yeah, good question. Does that make sense? I mean, it's a little weird, but again, we think that this was probably, here's what we suspect. Evolutionary wise, we think this was probably the first type of metabolism when cells were first learning how to do this process. So they were opening, they were pulling apart, they were processing one side, and things were going good. And then as cells started to get more advanced and started growing faster, they needed more energy. So then they started developing more enzymes as a rate of need. So through evolution, through demand, they started getting better processing, which led to an evolutionary curve to this side. When eukaryotes came along, we need a lot of energy. There's no way we could satisfy with just 1 ATP. So we always advance with this side. So that's the difference. So when you're looking at these, if I was to ask you about glycolysis, if I said which 2 pathways of glycolysis would give me energy potentially, which 2 pathways would they be? EMP and the ED. If I said which pathway is going to give me nucleotides? The EPP. If I said this is the pathway that most prokaryotes do and all eukaryotes do, which one would that be? EMP. If I said this is the few prokaryotes do, the ED, all right? So I don't spend too much time on these, I'm just showing them to you so that you understand the background behind it. We will continue to move on and focus on EMP because this is one that we're mostly familiar with, and this is also the one that most cells will use, so it makes most sense. But I want you to be aware that these are out there. So when we start talking about the process of respiration and using and developing more energy in the cells, that's going to be based on using that EMP pathway. Now, we're going to learn that some organisms that are aerobic are going to do aerobic respiration, which means that their final electron acceptor, what that means is that the element at the end of their pathway is going to be oxygen. And so what that means then is that oxygen has to be present to be able to do an aerobic pathway. Anaerobic bacteria or organisms who live without oxygen, instead of having oxygen at the end of their pathway as their final electron acceptor, are going to have something else. Now this could be sulfur, this could be hydrogen sulfide, this could be nitrates, something else. It's not just pure oxygen. Okay, so it has to be something where the oxygen's bound up or other elements are involved. So for us, we're going to learn that we need to have oxygen in place because we're aerobic respiration. Some bacteria out there are not, so they're not going to need to have their oxygen in place. We'll explain why. It has to do with the electron transport chain. So, features that are going to happen in the next steps of respiration. That pyruvic acid that we created going through the EMP or through the ED is going to get oxidized completely. So what does oxidize mean again? Losing electrons, yeah. So it's going to be completely broken apart. And we're going to get rid of some of those extra carbons that are present. And anytime we break carbons off, they leave the body or the cell as CO2. Okay, so CO2 is always going to be something that is going to be created when we break the carbon off. Now we're going to create some Uber drivers, we're going to create some NADH and FADH2s. Those are going to go on to another part of our respiration process. If we are doing aerobic respiration, we need oxygen to be at the end of the process. If we're an anaerobe, it can be anything other than oxygen. Alright, so an anaerobe can be, like I said, sulfuric acid, it can be anything. We're going to find that we're going to make most of our ATP in the electron transport chain. Now, for us to be able to get to the electron transport chain, we have to have oxygen present because it's our final acceptor. If I don't have oxygen, I get stuck and I can't move forward. So, the stages that are going to occur in our respiration process. That pyruvic acid that we created is going to go through a conversion step. This is sometimes called the preliminary reaction. This is sometimes called the conversion step. Different names. All it means is it's going to be converted from a 3-carbon molecule to a 2-carbon molecule. So the reason for this is because of the Krebs cycle. The Krebs cycle is the next step in our respiration dance. Okay, we started with glycolysis. The next step is the Krebs cycle. But to get there, I have to become a 2-carbon molecule because the Krebs cycle has a bouncer that will only accept 2-carbon molecules. So my pyruvic acid that has 3 carbons cannot progress. It has to become a 2-carbon to be allowed to get past that door and enter into the Krebs cycle. Okay, so that's going to be something that we're going to see. Now, when I change from a 3-carbon to a 2-carbon, I change my name. So I'm going to go from pyruvic acid to my 2-carbon molecule, which has the name acetyl-CoA. Okay, so that's where some of these weird names are coming from, because we're changing our carbon. Now, again, that preliminary reaction is also sometimes called the intermediate step, the conversion step, it's all the same thing. Okay, now we enter in the Krebs cycle, and once we get in the Krebs cycle, we're going to do some cranking around, we're going to be adding carbons, breaking them off, getting a little bit of energy, and then we're going to go to the electron transport chain. Yes, go ahead, Brandon.
So how does it get from pyruvic to acetyl-CoA? Is that the intermediate step?
That's going to be the intermediate step.
Okay.
Yeah, intermediate step, conversion step. None of anaerobic organism, I don't need oxygen present. But at the end of glycolysis, if oxygen isn't there for me, I cannot progress to Krebs or to the electron transport chain. So I can only make 2 ATP. I have to have oxygen present to be able to progress further. So I'll go through the intermediate step, and I'll get ready to go into Krebs only if oxygen's there. If it's not there, my enzymes are not gonna let me go, and I'm not gonna be able to go into the Krebs cycle. Okay, so just something to keep in mind. Okay, so here's our intermediate step that's gonna occur to our pyruvic acids. So what's gonna happen here, we have this 3-carbon molecule, which is pyruvic acid or pyruvate, they're the same. It's gonna lose one of its carbons, and when we lose a carbon, it always leaves a CO2. Okay, so it's gonna break off and leave a CO2. Now, because we're breaking a carbon off, that's gonna release some hydrogens. Remember, carbons are holding hands with each other, and they're holding hands with hydrogens. So when I break a carbon off, I release some hydrogens, which means that those can get added to an Uber driver. So I'm gonna generate a little NADH here. So my intermediate step is gonna generate a little NADH, not gonna generate any energy, not getting any ATP, just getting an Uber driver, and I'm gonna get a little CO2 kicked off. Now what's gonna happen then is enzymes are gonna help this process where I'm going to create a 2-carbon molecule, which is gonna be called acetyl-CoA. And acetyl-CoA being 2 carbons is allowed through the Bouncer's door to enter into the Krebs cycle. Alright, so what I want us to picture with this is that I have my pyruvic acid, let's say pyruvate, and remember I have 3 carbons and I have 3 carbons because I have 2 molecules of it, right? So I'm going to have to go through this intermediate step twice. Does that make sense? So I'm going to take this 3 carbon and I'm going to break it down to a 2 carbon. That's a very messy carbon. There. And I'm going to do the same thing over here. And that means I'm going to kick off a CO2 here, and I'm going to kick off a CO2 here. I release some hydrogens, so they're going to get picked up by my Uber drivers. So I'm going to get an NADH and an NADH. So now when I'm done, I have these 2 2-carbon molecules. Everybody with me on that? What do we call these?
Acetyl-CoA.
Acetyl-CoA. So here's my first acetyl-CoA. Here's my second acetyl-CoA.
The NADH is attached to the 2 carbons?
The NADH isn't attached, he's an Uber driver. So he's gonna come in and pick up the hydrogen and leave. So remember, NADH, how does he actually come in as? NAD. So NAD comes in, grabs a hydrogen and an electron, and it's gonna leave. And now it becomes NADH. And it's gonna go to the electron transport chain. He's gonna go somewhere else, which we're gonna get to. All right, good questions. So is this making sense to where we're at now? Okay, if you're confused, let me know. So I wanna make sure that we're ready to go on to the next step. That's why we break this apart. And then don't worry, we're gonna do a big recap and make sure that we go back to the beginning and carry us all the way through. Okay, so my acetyl-CoA is ready to be processed now in the Krebs cycle. So a lot of times what you'll read in a book is it'll tell you pyruvate from glycolysis is oxidized in the Krebs cycle. Well, sort of. Is pyruvate being oxidized? No, it's the acetyl-CoA. We've already done that intermediate step. Okay, so I put that in there, but just always remember it's already been kind of tweaked, so the pyruvate has become acetyl-CoA. Okay, we're going to get a little bit of ATP in the Krebs cycle. We're going to get a lot more Uber drivers, and that's kind of the goal, is to gather more hydrogens and electrons, because we're going to be taking those to the final 3rd pathway, which is the EGC. Now, the Krebs cycle is also a place where we'll break apart lipids and amino acids, and we can break apart purines and pyrimidines. And you might be going, well, what are those? Well, remember, a purine and a pyrimidine is a nucleic acid, right? So they're associated with DNA and RNA. Okay? So what this means then is when I ingest food, let's say I eat a salad. Let's say I eat lettuce. So I eat my salad. When when I'm adjusting that food, am I getting some carbohydrates? A little, there's a little sugar in lettuce, right? Am I getting any fats? Maybe if I have salad dressing, but yeah, probably not too many fats. Am I getting any nucleic acids? Yes. Is lettuce a living organism? So is a plant. So every time I eat, I know it's a weird thing, but we're getting nucleic acids. So when we're ingesting food, it's coming in with nucleic acids. They're not going to get broken down in glycolysis because that's sugar. Instead, they're going to get moved right to the Krebs cycle. Okay, same with fats. Now, have you ever heard of diets like Atkins or low-carb diets? Okay, if you know anybody who's ever been on Atkins or a low-carb diet, one of the things everybody will tell you is the first few days are the worst. You guys heard that? They call it the low-carb flu. Have you heard that? Like you feel tired and exhausted? The reason for that, you have decreased your sugar intake. So you are bypassing a lot of glycolysis. Instead, you're jumping right into Krebs. So we're actually making less ATP when we initially start a low-carb diet. Now, our body will adjust by creating more mitochondria, which help with the electron transport chain, and then it adjusts how it's using its sugars. It'll also start breaking sugars down that are being stored in the body. So that's the whole point. That's why you start burning your lipids. It's because your body is pulling sugars that are being stored. Isn't this cool? It's like kind of neat. It's like closing the circle, right, with nutrition. Okay, so what's going to happen here in Krebs is we're going to do a lot of rearrangements of our pyruvic acids that have been converted to acetyl-CoA and lipids and anything else that comes in. So Krebs is kind of like a little grinding wheel, so to speak. The thing about the Krebs cycle that amazes me is it was discovered by Hans Krebs, and it was discovered like in the late 1800s, early 1900s, which is just incredible to me that they were figuring this stuff out before they even had these high-powered microscopes and all these special ways to do it. It's kind of cool. Now, sometimes the Krebs cycle is called the citric acid cycle. And the reason for that is because the first product made in the Krebs cycle is citric acid. So it's sometimes called the citric acid cycle. I put that up there because I had a couple of students who did great in my microbiology class. They later on went on to take a physiology class at Santa Barbara City College. And they came to visit me during office hours because they were confused on metabolism. And they said, "You explained this so well, can you explain it again?" And I said, "Yeah, what are you confused on?" And they said, "We don't know what they're talking about with the citric acid cycle." And I looked at them and I go, "It's Krebs." And they went, "Oh." So they heard a different name for it. It was the same cycle. As soon as I said Krebs, they were like, "Oh, we get it. Now we feel foolish. We thought it was this different wheel." It's like, no, it's the same thing. So that's why I show you the different names. So that way you're aware that there's these different terms out there. It's also called the TCA cycle. So that's going to be for tricarboxylic acid cycle, which means that was what it was named when we thought the 3 carbons were first going in. We realized later acetyl-CoA is going in, so we kind of have moved away from that name because we realized it's not really a tricarbon, it's actually a duo. Okay, just throwing those names out. So here's the process, and again, it looks scary, but I will tell you right now, you are not going to have to name all of these intermediate products that are in there. So I'm not going to have you memorize every turn of the wheel. When we start learning what's going to be on an exam, what I really want you to know is know what's going in and know what's coming out. That's really what I care about. The reason we go through the process is just to make sure that you understand it. I don't want it to be like, a black box, like where it's magic and you don't know how it happens. So we go through the process. But I will never ask you to draw carbons or draw phosphates. To me, that's not, I don't care. I really want you to have a better, just a general understanding. Okay, that's really what my goal is. Because if I go in here and I start asking you to name all these steps, that's great. But are you ever going to be in a situation where you need to memorize every step and realize, no, you're never going to have explaining this to a patient, you don't care. But if you can give them an overview of how metabolism works, like, hey, this is how a low-carb diet works, that's kind of cool. That's my goal. Okay, that's what we're going for this. So with the Krebs cycle, we have 2 acetyl-CoAs that are ready to enter. And the first acetyl-CoA comes in. And the first thing that happens to our acetyl-CoA when it comes in, how many carbons does it have? Yes, 2. So the first acetyl-CoA comes where the number 1 is. Coming around the circle is a 4-carbon chain. Alright, so my 2 carbon just very innocently enters, and this 4 carbon comes around and goes bang and hits into it. And I create a 6 carbon chain. And guess what that 6 carbon chain is called? Citric acid. Oh, that's why it's called the citric acid cycle. I got it now. Okay, so I've made the 6 carbon chain. Okay, does that matter? No, it's just explaining to you where citric acid comes from. Think about this though. I just did all this work to take glucose, which had how many carbons? 6, down to 2, and now I've just gone back to 6. I just undid all my work. So now I gotta do this again. So what's gonna happen here is in this cycle, I'm gonna start rearranging and getting rid of carbons. Now every time I rearrange a carbon, when I say rearrange, what I mean is sometimes we're creating double bonds. Okay, what's a double bond? Well, that's when carbon goes from holding another carbon and a hydrogen to letting that hydrogen go and hold both ends of that carbon. Alright, that's a double bond. I'm holding on to the carbon twice. If I let that hydrogen go, who can grab it? NAD or any Uber driver. Okay, so we're going to see some Uber drivers being created in this cycle anytime a carbon changes how we attach to another neighbor. Okay, that's going to be our goal. Okay, so we have citric acid that starts with 6 carbons. The first thing we're going to do is we're going to start rearranging that citric acid. And as we start to rearrange it, we're going to get rid of one of the carbons. We're going to go, oh, I don't want this carbon. I went from 6, I want to go down to 5. If I take a carbon off, what does it leave us? What does carbon always leave us? CO2. Yeah. So I can kick a carbon off, it's going to leave us CO2. That's why it's showing CO2 there. If I kick carbon off, he's no longer holding hands with hydrogen. So who can grab it? An Uber driver. So I create an NADH there. Okay. Now I'm down here. I really wish I had my meter stick in here. I don't know where my meter stick went. I keep looking around. Where is this ridiculous student? I don't know. Where's my meter stick? Here's my meter stick. Yes. Now I feel like a teacher. Okay. Like the nuns used to. Okay, here we go. All right. So now we're here. Everybody answer the questions. I'll get your knuckles. Funny story. My stepbrother got kicked out of Catholic school because he was asking way too many questions. And his little knuckles were so broken, we had to buy 4 rulers. They broke 4 rulers over his hand. This is back in the old days where kids would listen to he had ADHD. We didn't know it at the time. Kids in my generation didn't have ADHD. You just sucked it up and sat there and be quiet, or you got hit with a ruler. Yeah. Thankfully, the times have changed. My daughter has ADHD. We kind of laugh about that. Like, my uncle or my brother will tell her, like, you're lucky kid. There'll be rulers in your future otherwise. Okay, so here we have NADH. And the reason NADH got involved here was because we popped off a carbon and we're now down to a 5 carbon chain. Okay? Now what's going to happen is we're going to get rid of another carbon. So when this carbon leaves, we go to a 4 carbon. Okay? When that carbon leaves as CO2, we release another hydrogen, we get another Uber driver. Now what's going to happen here is this 4 carbon is going to come swinging around the corner here. And when it comes swinging around the corner, it's kind of like if you picture a boat on a lake. If I'm a boat and I curve like this, what do I create behind my boat? A wave. I create an energy wave. So this energy wave, as this guy wings around, is going to be just enough energy to attach a phosphate onto an ADP that's nearby. Now our cells know this swing is coming on this corner, so they send ADPs and free phosphates here waiting for that energy. Okay? So I swing around, my energy wave creates 1 ATP here. Okay,
Where does the
great.
carbon come from again? Sorry.
The carbon is coming from rearranging these molecules. So see here I have 1, 2, 3,
Yeah.
4, 5. I pop off that carbon, now I have 4.
Yeah.
So it's just being broken off.
Okay.
The way you can kind of picture carbons, when I was taking OChem, I kind of picture this. Do you guys remember snap beads? There were these beads you could snap together and you could pop them apart. That's kind of what they are. It's like you kind of picture them sticking together and then they separate.
Okay.
And they have no problem letting go. Enzymes come in and like break them off all the time.
But where's the one that creates the wave come from?
Oh, so the wave is this 4 carbon molecule, and enzymes are swinging them around to go to the next part of the cell.
Oh.
It's all happening in like a circular motion.
Okay.
And so it's just like if I grab somebody and swing them, it's just creating an energy wave.
Okay.
So that's not from a carbon, that's just from momentum.
Oh, gotcha.
Yeah, so I'm just swinging. So, and when I swing around here, holding onto a hydrogen to letting go and grabbing another carbon's hand. So they're gonna go grab another carbon's hand. When that happens, I'm gonna release a hydrogen, okay? And then I'm gonna see that I'm gonna do that again. I stay in my 4-carbon state, but I create another bond which releases another hydrogen. So when I'm done with this, I've created 1, 2, 3 NADHs. And that's from either carbons letting go or carbons grabbing double bonds. Okay, grabbing another carbon's hand. Okay. I also created a new Uber driver who's entered in the building, and that's an FADH2. Alright. Now, this guy's job is the same as an AD. It just picks up hydrogens and it's gonna shuttle them. These are just Uber drivers. Okay, and they're all gonna be involved with this process. So, the big picture, I come in as a 2-carbon. Oh, here, I should do this in. I come in as a 2-carbon. What comes swinging around? A 4-carbon. Okay? I smack into each other. I've created a 6-carbon. What is this one called? Citric acid. That's the only one I want you to know in this cycle, is citric acid, because that's another name for it. Okay. Now I have citric acid in place. That's this guy here, 6 carbons. I'm going to go through, I'm going to pop a carbon off. When I pop a carbon off, what do I release? CO2. And what else gets released? Hydrogens, who are going to get grabbed by an Uber driver. Okay, so I created an NADH. Now I move down here, I'm going to pop another carbon off. What do I create? CO2 and another hydrogen, which gets grabbed by NADH. I come swinging around the corner. What do I create down here? ATP from that energy wave. Okay? I swing around, and now once I'm on this side, some enzymes are going to tell my carbon to let go of another hydrogen and create a double bond. So I go from holding hands with a hydrogen to holding double hands with a new carbon. When I do that, I release that hydrogen and I create a ADH. Alright? So I have another Uber driver. Up here, I do the same thing. Another carbon, one on the other end lets go of the hydrogen and double hold another carbon. So now I create an ADH. So when I'm done with this process, how many NADHs have I created? 3. How many FADHs? 1. How many ATPs? 1. Excellent. So that is what I've created for my first acetyl-CoA. How many acetyl-CoAs do I have? 2. So I'm going to do this process twice. So the first acetyl-CoA comes in, cranks through this process, and I get 1, 2, 3 NADHs, 1 FADH2, and 1 ATP. But I'm not done, because I still got the second acetyl-CoA, who's waiting for his turn. Now he comes in, and he goes through this process. So when we're done with this process in total, what we're going to see is that we're going to end up, well, I don't have it on the slide. We're getting, when we're done with this process in total, so at the end of Krebs cycle in total, we're going to have 2 ATP, 6 NADH, and how many FADHs? 2. Okay, so that is our end step. So on an exam, you want to make sure, is the question asking per acetyl-CoA? Because if I say per acetyl-CoA, what is my total? 1 ATP, 3 NADHs, and 1 FADH2. That's per acetyl-CoA. But if I say we're looking at the end of the Krebs cycle in total, or we're looking at the end of the Krebs cycle at the end of the process, what is my total? This. Yes, the 6, the 2, and the 2. Alright, so this right here is my final end result. So you always want to read your exam questions to make sure, am I asking per acetyl-CoA or am I asking in total? Yes.
Okay, so just to clarify, in terms of looking up for the exam,
Yes.
know what comes at the end and
Yes.
understand how you get citric acid.
You got it. You got it. So know what goes in and what comes in at the end. So that's going to be the process for everything. So just to recap, to make sure we're all on the same page, let's talk about BMP again. What goes in?
Glucose.
Glucose. Okay, so I have glucose going. What comes out? wants a 2-carbon chain. So acetyl-CoA has a 2-carbon chain. It can get to the bouncer. If I try to go in as a 3-carbon, I'm too big. It's not gonna let me go in. Okay, so now I go into the Krebs cycle. So what enters? Do they enter together?
One at a time.
One acetyl-CoA, yeah. So one acetyl-CoA is what's going to enter. And as soon as it enters, and I'll put over here 2 carbons, it gets hit and it becomes a 6-carbon. What do we call that 6-carbon? Citric acid. Yeah, so I just want you to know that because again, that's a term that's used interchangeably. So one acetyl-CoA comes in, it turns into citric acid. Okay, I don't care about knowing oxalatic acid. I don't care about any of that, so we can stop at this point, all right? What I want to know is what comes out. Now, what comes out per acetyl-CoA is going to be right over here. 1 ATP, 3 NADHs, and 1 NADH2. At the end, because I have to go through this twice, I get this result. Is that feeling a little better? And we'll keep going through this, because believe me, I've tried a bunch of different ways. I've tried shock and awe, where we went through all of it, and then we went back, and I feel like this is better. We're kind of waiting. We're kind of waiting, and we're kind of working our way through. Again, I'm not making you memorize every single step, because who cares? I want you to know the big picture. Because as long as you understand the big picture, you're understanding the process. To me, that's more important than being able to name every single chain in between. Okay, the next thing to know about the Krebs cycle is where it occurs. So the Krebs cycle is going to occur in the cytoplasm, of our bacteria or of our prokaryotes. Okay? So when we're talking about Krebs, it's going to occur in the cytoplasm of our prokaryotes. Now, for us, because we're a little more sophisticated, I don't like to brag, but we're more sophisticated, we've got organelles. So what we do is we do this process inside of an organelle. Remember when we talked about our different organelles, we talked about the mighty mitochondria? So for us, we do this in the mitochondria. Now, hopefully this makes sense, because where do we think the mitochondria came from? A bacteria. Yeah. So bacteria do this process in cytoplasm. We ate a bacterial cell and made him a mitochondria, so we do this in the mitochondria. Does that make a little bit of sense? Now, backing up one more step, for glycolysis, glycolysis is going to occur in the cytoplasm for everybody. That's going to occur in the cytoplasm for eukaryotes and prokaryotes. Just to back up a step, all right? So, cytoplasm and cytoplasm or mitochondria, depending on who we're talking about, all right? Okay, so here is a simplified version of the Krebs cycle. Again, I will never ask you to draw this on a test. I will never ask you to tell me which step is an NADH made. I don't care. I just want you to know what the net totals are, what's coming out, all right? But I am showing you this so that way you can kind of follow it, and as you're learning it, it starts to make more sense, all right? And what this is really showing is it's showing that we're starting at the top from acetyl-CoA, who's a 2-carbon, who gets rammed into to become a 6-carbon, and as we go through, we're just rearranging and releasing carbons to get those Uber drivers built. So when we're done for one glucose, so this is a good way to know how I might ask this on a test. Per glucose molecule, what are my totals? Per glucose molecule means that both acetyl-CoAs have been processed. Okay, so per glucose molecule. I'm going to end up with 6 NADHs, 2 FADH2s, and 2 ATPs. Now, you might have noticed in my notes, I sometimes have FADH2, I sometimes just have FADH. That's because I don't care. You don't have to put that 2 on as far as I'm concerned. So, in organic chemistry, when we have to keep track of our hydrogens, you do. If you put FADH on test, I'm not going to go. All right, I don't care. You don't have to have it. Yeah.
So you were saying, like, for an exam, this question is specifically for one glucose cycle, specifically for the Krebs cycle, not just.
If I just said per glucose, Glucose means the glucose has been processed completely, so we're looking at the end of the Krebs cycle.
So that includes all the other processes.
You got it, you got it, yep. So everything that's led up to this point. So that's what we're looking at for the Krebs cycle. All right, is that making sense so far? Yes.
Shouldn't we have 4 ATP then?
So if we added in glycolysis, yes, but this is saying just the Krebs cycle.
Just the Krebs cycle, okay,
Just the
got it,
Krebs
okay,
cycle.
got it,
Per glucose in the Krebs cycle.
okay, got it.
Now, if I said in total for glycolysis and the Krebs cycle, where would I be? You're right, I'd have 4 ATP. You're right, yeah. Because we want to add those two nets from glycolysis. Good thinking, I like what you're thinking there. Everybody doing okay so far with this? Okay. So the next step that we get to is the electron transport chain. So the electron transport chain is where we really finally get a big bang for our buck. We've been doing lots of work. I don't know about y'all, I'm exhausted. We've been processing the sugar, we've been cranking them around, we've been taking hydrogens, and at this point in time, I'm walking away with a measly 4 ATP. That is not worth my effort, I'm worth more than that. So now, I'm finally gonna get the big bang for my buck by going through the electron transport chain. Now, again, to do this process, I need to have oxygen present for those of us who are obligate aerobics. So for us, we have to have oxygen present to do Krebs and to do the electron transport chain. Glycolysis is the only one that can be done without oxygen for us. Okay? Alright. Now the other thing to keep in mind is when we make ATP here in this stage, it's referred to a lot of times as chemiosmosis. And the reason it's called that is because we're going to learn that we're going to be moving molecules across a membrane, and that's going to create some energy. So that's why it's called chemiosmosis. Osmosis means moving across a membrane, and so we're going to be moving molecules across the membrane. You're going to see that. Okay, so up to this point, we have been loading up Uber drivers. NADH is ready to go. FADH2 is ready to go. My Uber drivers are full. Well, what they need to do next is they need to drive to the end location, which is the electron transport chain, and they need to drop off their passengers. Their passengers with them, remember, are hydrogens and electrons. Okay. So when they get to the electron transport chain, the electron transport chain is going to have molecules there that as soon as the Uber drivers pull up, kind of like pull up to a curve, they're going to grab the electrons and hold on to them. And the hydrogens who gave the electrons up are going to get pushed to the side. Okay? It's kind of like if you've ever gone to a party with your friend who's way more popular than you. You might walk through the door together, but then they get grabbed and taken off to the main part of the party, and you're just kind of standing there going, well, I guess I'll go get some food. You know, because you're not getting grabbed and pulled into the mob. The electrons are the party people. They're getting pulled into the mob, and the hydrogens are kind of like the +1. They're the accessory that's going, now what do I do? Okay. The other thing we're going to learn about our hydrogens is hydrogens can be a little fickle. Hydrogens are never happy. And what I mean by that is when they have their electrons, how happy do they feel? Are their shells full? No. So they willingly give their electrons up. But then, have you ever had giver's remorse? Where you gave something away and then wish you hadn't? Yeah, I have this. My little cousin is up in Cal Poly. She came to visit me, and I still have this, giver's remorse. I had a sweater out, and she's like, oh my gosh, that's the cutest sweater. I go, yeah, I hardly ever wear it. I should probably donate. She goes, I'll take it. And now every time I see her in my sweater, I'm like, I shouldn't have done that. I should have kept my sweater. I have total giver's remorse. That's what hydrogens do. They do the same thing. I feel it. They give up their electrons. They're like, yeah, you're right. I don't want this extra guy. I don't want to try to find another one to fill my shell. You're right. You talked me into it. I'll just give them up. You take them. And then as soon as he gives the electrons up, he's like, ooh, I kind of wish I kept that electron. I kind of miss it. We had some good time. We should have hung out more. So now the hydrogen is kind of wishing it had its electrons back. And that's what's going to drive the electron transport chain. We're going to separate those hydrogen ions and not let them have their electrons. But, kind of like my cousin does to me, we're going to make sure we show them the electrons. She wears the sweater around me purposely. I know this, just to show it off. And all the key ways that she can accessorize it. Like, yeah, same idea. So they're he's showing the hydrogen ions the electrons. They're like, woo, here go your electrons. And the hydrogen ions want them, but they're not going to let them have them until they get to a special enzyme. This enzyme is going to be a special protein channel that the hydrogen ions are allowed to move through. And we're going to learn that that channel is called ATP synthase. Ooh, that's a good name. ATP synthase. What does that sound like it's making? ATP, synthesizing ATP. So we're going to let the hydrogen ions move through and reclaim their electrons, but only when they pass through ATP synthase. So that's going to be our process. So we're going to push the hydrogen ions away from the electrons against their gradient, and it's kind of like pushing them uphill, and then we're going to let go and let them fall. And when they fall through this ATP synthase, as they fall, just like we saw in the Krebs cycle, we're going to generate some wakes, some waves of energy, and we're going to be able to attach phosphates to ADP. That's going to be our goal. Okay, so let's look at what the process is going to look like. The first Uber driver to arrive is NADH. NADH comes in, and it passes its electrons on to an electron acceptor. Do not memorize the names. Just know an electron acceptor takes it. That's all I care about, okay? My electron acceptor is going to grab these electrons, and it's going to pass them to another electron acceptor, and so on, and so on, and so on. The idea behind this is my electron acceptors are basically chemicals that are also wanting to fill their electron shells, okay? So they grab an electron, and they're like, yes, my shell is full. But right beside them is a bigger, badder molecule that wants that electron. And because he's bigger, he's going to pull the electron towards him. It's kind of like the bully at lunch that would take their money. Same idea. You know, they come there like, I'm going to take that. You're like, yeah, you can have it. Whatever. I'm just going to stay here. So they pass their electrons along. So in this chain, each acceptor in this chain has a bigger pull towards those electrons. And we're going to learn for us, the biggest, the baddest electron grabber, the one who never has enough electrons, who's always hungry, oxygen. The other way you can kind of picture this, picture that you're holding like a sheet, okay, like a bed sheet. And you have 4 friends with you, so each of you is holding a corner. And then I come in with a bowling ball and throw it on that sheet. What's going to happen to your sheet? It's going to go whoop like this. What happens now if I put a ping pong ball on the edge of the sheet? Where's it going to go? Straight down to that bowling ball. Guess who's the bowling ball? Boom, baby, oxygen. All right? So oxygen has what we call electronegativity. He's pulling the electrons towards him. That's basically what's happening. He has a lot of force, like a bowling ball on a sheet. He's pulling the electrons toward him. Now this is also why if we don't have oxygen, this whole process shuts down. Imagine that, again, your friend's holding that sheet, and this time there's not a bowling ball there. And I put a ping pong ball on the edge. Is it going to go anywhere? No, because your sheet's flat. So I have to have this guy in the center for us as our final acceptor to pull the process along. If I don't have oxygen in the middle of that sheet, nothing's getting pulled towards it. Okay, so this is just kind of a way you can kind of visualize what's happening. So my first Uber driver arrives in ATH, it drops off its electrons. Its hydrogens are going to get pushed on the other side of this membrane boundary. Okay? Now, we're going to learn that this membrane boundary for us is inside of the mitochondria. Okay? Ooh, the prokaryotes don't have a mitochondria. So what do you think they might be using for their membrane boundary? They're in the cytoplasm for Krebs. What's something that's around the cytoplasm that's a membrane? The plasma membrane or the cell membrane. So they're going to do this process at the plasma membrane or the cell membrane. All I need is a layer, because I need a layer to have the electrons on one side and to push the hydrogens on the other. Now the reason they can do this, they can push the hydrogens outside of their plasma membrane and not lose them, is because what is outside of their plasma membrane? A cell wall. So those hydrogens are not leaving. in that little interspace, but they can't leave. If we tried to do that, our hydrogens would be like, peace, ma'am, see you later, and they'd leave and go to another cell. So we can't risk that. We got to do this in the mitochondria. Okay, is this kind of making sense how we're going with this? Okay. All right. Any questions so far? Okay, so my NADH comes in first and drops off its electrons. Now, because NADH comes in first, the electrons from NADH are going to travel a little farther. They get picked up by the first electron carrier as opposed to FADH, who comes and drops off later in the process. The way you can picture this, picture like a taxi dropping off in an airport. NADH is the first taxi that pulls in and he parks in a spot and drops off his passengers. FADH goes around and parks farther up the curve. That's basically what's happening. He drops off his passengers. Okay, he has to go around NADH and park. The only difference with this then is since NADH has its electrons travel a little farther, it gets a little more ATP for each hydrogen that it brings in. My FADH, because it comes in later, it's only going to generate 2 ATP. So this is just the difference between the two. So that's something that could be on a test question. How many ATP per FADH? 2. How many ATP per NADH? 3. Yeah, so right there you have that. You can show that. Okay. Now, when I drop off my electrons, again, it's a game of hot potato. They get passed from acceptor to acceptor until they get down to the bad boy of oxygen, who's at the very bottom, who is ready to grab them. So again, I need these guys to pull that layer towards them. Now again, as I said, this is going to happen in the plasma membrane for our prokaryotes. It's going to happen for us in the mitochondria, in an inner membrane. So we just need to have a membrane to push hydrogens to one side and to build up a gradient. Okay. So my electrons are getting passed, my hydrogens are on the opposite side, and I'm building up the process to get ready to make a lot of ATP. Yes.
So I just wanted to make sure that I understood.
Yes.
So we have NADH, and NADH is carrying a hydrogen atom. So I gotta see like the electron affinity of these like channels or whatever is attracting the hydrogen atom. They spit that hydrogen out there,
Yep.
take the electrons, and then I guess because the other channels have like more affinity, the electron like gets passed through, so it ends up at oxygen.
Yes. Yes. So the idea behind what she was just saying is just, she was like, I want to make sure I understand. So NADH arrives, there's an electron acceptor right there who grabs the electron, who doesn't want the hydrogen's the plus one that nobody cares about, so he shoots the hydrogen away. He goes on the opposite side of the membrane. So now this electron acceptor has these electrons, he's happy. But right beside him is another bigger, badder electron acceptor who wants that electron, who's going to pull the electron towards it. And then right beside him is another bigger, badder electron who pulls the electron. And they just keep going down this chain. Who's the biggest badass waiting for us at the end? Oxygen. He's down here. He's like the Arnold Schwarzenegger. He's like, send them to me, I'm down here. Send them. He's waiting, and he grabs the electrons. All right, so now we have the electrons who have been passed from one acceptor to the next. And now oxygen is holding these electrons. He's super, super happy. But our hydrogens who want their electrons back, just like I wore my sweater from Harlem, who want their electrons back, are going to try to figure out a way to get there. And guess what? We have a way for them to get their electrons back. The hydrogens who have been sitting up here really, really having that giver's remorse are going to come to an area where there is a protein channel, a way to get across the membrane. And this protein channel is like ATP synthase. So I'm going to let the hydrogens start to fall through this channel. And as they fall through, the hydrogens are going to bind with oxygen. Remember when we said we made water as a waste product? This is where our water is coming from. The bond with the oxygen and they're creating water, okay? Our cell doesn't care about that. It cares about the electrons. The electrons are here, the hydrogens come through and grab them, but here's the beauty. As they fall through, they're generating wave after wave after wave of energy. And this enzyme, this protein channel that's sitting there, it actually starts to spin like a turbine, like a water wheel. If I push water over a wheel, how it starts to spin, that's what it starts to do too. And as it spins faster and faster, as these hydrogens keep falling through, it's generating energy waves. And those energy waves are enough energy to start attaching phosphates onto ADPs. So this thing, like I said, it will actually spin and it'll attach phosphates to ADPs. And I'm going to generate a boatload of ATP. Finally, I'm getting a lot of bang for my buck. I'm finally getting a lot of ATP here. So what's going to happen then is those hydrogen ions are going to pass through that ATP synthase. They're going to cause it to spin, and I'm going to make ATP. So I have ADP sitting there waiting. I have phosphate sitting there waiting because the cell knows this is going to happen. It has them sitting there just waiting. And then as soon as this energy starts coming, we start stacking them together. Okay, and we start making our ATP. So that's what we see here. Now, I don't care about knowing how many hydrogens that 6 fall through at a time in a I don't care about that. Just know hydrogens are falling through, binding with oxygen, and that falling through is what's generating energy to create ATP. Yes?
That being said, you said each NADH will make 3 ATP, each FADH will make 2 ATP.
Right.
That has to do with the amount of electrons that are flowing through the intermembrane space.
Correct. It has to do with the amount of energy that behind the electrons as they're passed on. Think about it this way. With a water wheel, if I drop water at the top of the wheel, it generates more energy than if I jump in the middle of the wheel. So FADH is coming in after that wheel is kind of moved, so it doesn't generate quite as many electrons of energy as the hydrogen we started earlier. That's really what it is. Yeah, good question. Okay, so with my ATP synthase, this is a special molecule. It ends in ASC, so what does it mean that is? It's an enzyme. It's a special enzyme. It's going to allow my hydrogen ions to flow down and reconnect with their electrons. Now, when they reconnect, as they flow down, they start to spin the ATP synthase. As they're falling through, they're creating energy waves like the wake of a boat. And that's going to be enough energy to cause ATP synthase to spin and generate waves to attach phosphates back onto ADP. When I do this, attaching my phosphates back to ADP, I make some ATP. This is where it gets really cool. For all of my electrons that are brought in, after I account for all of my NADHs and all of my FADH2s, at the end of the electron transport chain, I make a total of 34 ATP. Woo! How many have we gotten in the Krebs cycle? 2. How many did we get in glycolysis? That was nothing. I want 34. Yeah, that's what I'm talking about. So now I'm getting 34 ATP. This is awesome. So at the end of this process, at the end of metabolism, at the end of breaking down a glucose molecule, my prokaryotes walk away with a total of 38 ATP. Us eukaryotes walk away with a total of 36 ATP. Now you're probably scratching your head. I know when I was a student, I was like, what the heck? We got gypped. So let's first of all figure out how we got 38 ATP. I generated 34 in electron transport chain, but I had 2 stages before. How many did I get in glycolysis? 2. So 34 plus 2 gets me to 36. How many did I get in Krebs? 2. So that gets me to 38. That's where the prokaryotes are getting their 38 total. What happened to us? Okay, let's think about this though. We moved our process from the cytoplasm to where? To the mitochondria. We have to pay a penalty. Moving from a cytoplasm to the organelle, it costs us a little energy. So we end up with a total of 36 because we pay to ATP to go into the mitochondria. We have to pay a little bit to transport. We pay a little fee to shut. So our tokens, our Uber drivers, they have to go farther. So we have to pay for their gas, so to speak. So it ends up counting us losing to ATP. So we get a total at the end for every glucose molecule of 36 ATP. The prokaryotes, they get the full 38 because they're not moving anywhere. They're staying right in one place. They're doing cytoplasm, plasma membrane. They don't have to go to an organelle. Okay, so we pay a little penalty because you have to go to an organelle. Okay, at the end of the process, the final electron acceptor is the one who's really dictating how well this is going to flow. Who's our final electron acceptor? Oxygen. Excellent. So if oxygen is not present, Krebs and the electron transport chain will not occur. We get stuck in glycolysis. All right? And this can happen whenever we have low oxygen levels. Have you ever worked out so hard that your muscles were sore the next day? You got stuck in glycolysis. You didn't have enough oxygen coming in. And we're going to talk about that process next time we're together. Because we can get stuck in glycolysis, and when we do, we have some problems. So we're going to stop here. I'm going to end on this slide, so I think it's a good slide to stop on. Next time, we're going to go through this whole process one more time on the board and go through it together. Y'all are going to